In this video, we will be talking about how bacteriophages can be used to transfer genes from one bacterium into another. Let's get to it. So first, we need to talk a little bit about what bacteriophages are. They are viruses that use bacteria as their host. Now, there are many different kinds of bacteriophages. We're going to be talking about one particular kind that's called T4, and this is a model of what these bacteriophages look like. This is an actual picture from an electron micrograph shown here. Now, a bacteriophage is going to have a number of proteins that make up the entire bacteriophage. For instance, there is a head of the virus. That this is a protein coat with a certain sort of geometric shape, and then it is filled with DNA. Oftentimes, this is phage DNA, but as we're going to see, it's not always filled with phage DNA. There's also a portion called the tail right here, and then there are tail fibers, which are sort of like little legs. And these are the major components that make up a phage. Now, the exchange of DNA from bacteriophage, and in fact, the way bacteriophage like T4 reproduce is called transduction. So we're going to go through what transduction looks like. The first stage in transduction is that the phage is adsorbed to the bacterial host cell. So those little tail fibers, they bind to components of the outside of the bacterium and attach. The next stage in transduction is that the phage DNA is injected into the host cell. That DNA then produces certain kinds of DNases that will chop up bacterial DNA. So the host chromosome gets chopped up as well. The next stage, the DNA of the phage is replicated, and then its DNA is read to create the protein components that make up more phage. So you get more heads, you get more tails, you get more of the tail fibers. The fourth phase, the fourth stage, let's say, is that the bacteriophages are then assembled, and those bacteriophages then take up some of the DNA that's within the cell, mostly phage DNA at this point. Of course, the last stage is that this cell is now loaded with a whole bunch of new phage, just entirely loaded, much more so than was illustrated here. This then causes the cell to be lysed, so it breaks open, and then phage then spills out, where those new baby phages can go around infecting other bacteria. This whole process is called transduction. Now, there are ways of assessing bacteriophage density, which is not unlike, it has a lot of similarities, at least, to the way that we can measure bacterial density in a liquid media. So let's walk through kind of how we would do this. So we're going to first have our initial stock of bacteriophage. It's grown in some kind of stock. And then we can make a number of serial dilutions. So the first dilution that we're making is just a 1 to 10 dilution. And then in this example, they're doing 1 to 100 dilutions after that. So we've got 10 to the negative 1, 10 to the negative 3, 10 to the negative 5, 10 to the negative 7. Okay, so you got a bunch of different dilutions. Now, what's different about how you assess bacteriophage density is that you do what's called a plaque assay. And the first thing that you need for this is that you will need some sort of bacterial, a petri dish filled with agar. But the difference for this petri dish than the way many biology students might normally see a petri dish is that it's going to be covered with bacteria, just literally loaded with bacteria. And we refer to this as a lawn. So the bacteria, like you won't be able to see individual colonies of bacteria. It is just completely covered with bacteria. And so then what you do is that you then either spray on, or you might, typically it's just like sprayed on, like spritzed on, or you put on some sort of volume of, and in this case, what we're putting on is a 100 microliters or 0.1 milliliters from several different kinds of dilutions. In this case, we're using 3 different dilutions. We're using the 10 to the negative 3, 10 to the negative 5, 10 to the negative 7. So you put this on, and now what we're looking for is not bacterial colonies. We're looking for how much of the bacteria is killed by the phage that is placed onto the plates. So we see, in this case, 3 sort of typical ends that are 3 typical outcomes that one might expect to see when you do this sort of dilution with bacteriophages. The first, using the higher dilution here, the 10 to the negative 3, when we look at the plate the next day, we see that there's absolutely no bacteria. All the bacteria are lysed, which means that the plaque, which is now entirely just phage, is completely covering the plate. So there's no bacteria. The lawn is completely destroyed. So we wouldn't see any of this sort of yellow muck on it. In the other extreme end, with the lowest dilution, when we put on phage, we just see a lawn of bacteria. We see no plaques. So there's no phage that actually made it on, because the phage by this point was just so diluted that there was basically none in the entire solution. In the middle dilution, what we see in this case is sort of like the inverse of when we were looking for bacterial cultures. What we're seeing here are little holes of missing bacteria. And instead, what you're going to see there are what are referred to as plaques. So you're going to see a bunch of phage. If you could look really, really closely, there'll be phage that's within each of these little plaques. So these plaques, as long as we can see individual plaques like this, they had to have been started by an individual phage. So when you're doing and you're assessing phage density, all you need to do is go through and count your plaques. And then from there, you can then calculate out, using this calculation down here, the total number of phage that you had in your initial concentration. So one thing that's kind of interesting about phage transduction is that it can result in bacterial DNA being transferred to other bacteria. So let's walk through this a little bit. So if we've got a phage here that is injecting its DNA, and it's illustrated in green here, the first step is then, that's of course the first step. After that, we get replication of the DNA of the bacteriophage. And of course, we also get fragmentation of chromosomal DNA. That, of course, is going to happen. We've got those DNases working on to break up the chromosomal DNA. Then you go through all those other steps that we talked about before with the transduction reproductive cycle of bacteriophages. But what can happen, and often does happen, is that some of these bacteriophages, in fact, take up bacterial DNA instead of phage DNA. And here's an example of a bacteriophage that is now loaded with bacterial DNA. So, of course, for those that took up the phage DNA, they go through and they do the same thing of creating more progeny phage. But these kinds of bacteriophages that have taken up bacterial DNA, they could be carrying genetic information. And in this case, what we see is that there's a gene for producing the amino acid leucine on this little fragment that is now packed into this DNA, and into this bacteriophage. This bacteriophage, if it infects another cell, instead of injecting bacteriophage DNA, it's going to be injecting this bacterial DNA carrying the leucine. And if this is a leucine that is a functioning leucine, and if it's injected into a mutant that is leucine negative, meaning that it is missing the gene or has a mutated version of this gene to allow it to produce leucine, what can happen is that you can then get recombination of this fragment of DNA, and it can be swapped out and actually repair the deficient gene. And in fact, scientists do use phage-mediated transduction to change the genome of bacteria in this way. Transduction also allows the transfer of genetic material into host chromosomes, including co-transduction. So again, this is this illustration of how you might use transduction to replace that leucine in the same way that I just showed. Of course, bacterial chromosomes have lots of other genes on there, and it might be that the bacterial chromosome, the portion, the fragment that was injected by the phage, could replace a gal, for instance, or it could replace the ability for bacteria to produce biotin. But if these two genes are actually close enough, you can actually get co-transduction, meaning that the fragment could contain both, say, gal positive and biopositive genes, and then in fact replace both of those deficient genes. And you can look for, when you do this kind of transduction experiment, you can then look for examples of co-transduction. And in fact, that can then be used to tell us information about the map of the bacterial chromosome. So in fact, we're going to look through kind of how one might go about calculating this. So if we wanted to determine co-transduction frequency of, say, two genes, we're going to just call them C plus and D plus, into a background cell that is C minus, D minus, and E minus, okay, just example sort of default genes. And if we select for C plus, we can look for co-transduction of C plus and D plus. So let's say if we're doing this and we find individuals, we count up our individuals and the kinds of co-transduction that we have seen, we may see, for instance, co-transduction of all 3 genes. This would be an indication that at the very least, that C, D, and E are close enough where they can end up in some fragments. We may see the genetic composition be that we might see C and D, but not E. And if that's higher than that, that might indicate that C and D are closer to each other than they are to E, than either of them are to E. Or at the very least that C and D is closer to each other than, say, D is to E. If we happen to see a lot of C's and we're selecting for C, that would make sense. This would mean that the C is kind of close to D, but it's not that close. And then if we want to look for C and E, but not D, and if we see that that number is really, really low, either 0 or close to 0, that's an indication that C is really not all that close to D, which would mean that C, that the order of the gene is probably C, then D, and then E. And so then from this kind of information, we can go ahead and count up all the individuals that we see. We count up the number of co-transductants for the gene that we're interested in, looking for, say, C and D, if that's what we're looking for. And you add up those two numbers, 57 plus 76, and you divide it by the total number of the transductants, the total number of individuals. And in this case, it just happens to add up to 500. And so you find out what that proportion is. If you multiply by 100, that's going to give you a percentage. And then that's going to then tell you the percent of co-transduction. That number is going to be lower if the if C and D is like closer to each other. Just it's sort of similar to how we can assess mapping with if we wanted to introduce genes into bacteria using transformation techniques. And obviously, this is a way to assess the randomness of where those DNA breakage sites occur due to the DNases of the phage. So this has been a short video explaining how we can use bacteriophage, understanding the bacteriophage life cycle, how its genome can be used to sort of obviously create additional phage, but that these bacteriophages also have the ability to take up bacterial DNA, and that we can in fact use this as a way to do mapping in bacteria. So I hope you've enjoyed this. If you have, hopefully you can just like and subscribe to the channel, and I will talk to you later. Thank you.